Hello everyone. So today we will understand linking number. In this video we'll try to understand what is linking number, and we'll also understand how the linking number is associated with positive and negative supercoiling. We'll also try to understand the superhelical density, and we'll also see how linking number is linked with the superhelical density. So all right, without any delay, let's get started. So I have, as per the general format of my videos, I have laid out all the important points. So we'll discuss one by one. On the top, you can see I have written the title, which is the linking number. And linking number is usually written as the abbreviation, which is L, capital L and small k. So this is how you write linking number. So capital L, small k. Next is a few points regarding the linking number. What is linking number? And illustration is also there. Linking number is nothing but the topological property of the double-stranded DNA molecule. So now what we are doing is we are trying to understand the topological properties of the double-stranded DNA molecule, not the single-stranded DNA molecule. I'll tell you why. Next is there is no change. When there is no change in the DNA molecule, no bend in the DNA molecule, and you need to have that condition is DNA need to be intact. Then we study the topological property, which is the linking number in that case. So as you can see in the illustration number 1, you have linking number 1 when these two different strands, we're just taking the hypothetical condition. In this case, DNA is not going to be like this only. It will have multiple turns and multiple helix in this region, and then that will constitute the circular DNA molecule. But just to understand how we define the linking number, so when one strand penetrates the other one, that we define as a linking number 1. In second case, you can see to make it more simplified, many textbooks, you will see many textbooks. Most of the data that I'm taking here is from the book Lehringer, and this is a fantastic book. You should read that book. If you want the reading material for you is for biochemistry, Lehringer. And then I'm taking that particular information and trying to explain it in my own way. So to simplify this, as you can see, one strand is kept just the circular strand here, that is the one, and the second one is being shown as the twisted strands. So it's very easy for us to define how many penetrations that are happening with the strand number 2, right? So you can see in this case we have linking number 6. Total linking number is 6. Why? Because you can see the penetration. So if I take this is 1 over here, and then this is second, and then this is third, and then this is fourth, and then this is fifth, and then this is sixth, right? So this is how it is happening, and then we can understand total numbers. Linking number in this case is 6. So if you have a structure and you have a question and where this structure is shown, you can easily define the total linking number of that molecule by just seeing how many times the particular strand is penetrating the other one, right? So you can see the typical definition of the linking number is how many times or number of times the twisting strand, in this case, because we are representing these two strands as one simple circle and other one is more twisted, and the twisting strand is penetrating the other strand. How many times it is doing that is the linking number you have, right? So this is the general overview of the linking number. Now let's move on to the more linked topics with the linking number and try to understand some of the advanced information on the linking number. All right, let's continue with the linking number. In the first slide, what we understood that linking number is the topological property, and we also find out how we can calculate the total linking number. It is the number that is being used for the number of times when one particular strand penetrates the other one. As you can see in this illustration here, we have I have simplified the structure, and now we have total 1, 2, 3, and 4 linking number in this particular structure. Right. Another concept in this case is the concept of topological bond. So as you can clearly see, what is happening in this case is one strand is moving along to the other strand, and then it is forming that twist, and it is forming that typical double-stranded DNA molecule. Now, just take the condition when you have no hydrogen bonds in that particular structure. Means when you have a particular double-stranded DNA molecule, but all the hydrogen bonds, they are broken. But what will happen in that case? Let me make that structure. I'm just trying to simplify what I've written here. So in case of double-stranded DNA molecule, we have these bonds are called hydrogen bonds, where two strands they are joined together. Now, for some reason, if these strands they are broken, what will happen? The bonds are not there, but these strands they are going to be like this. They are going to have that twisting around each other, and that we can define as the topological bond, because it will still be there. They will be twisting around each other, and although there is no physical contact between them, right? So that concept we can call it as the topological bond, where you don't have a physical contact, you don't have any bonding or hydrogen bonds between the DNA molecule, but still that topological bond is there. So that is the concept of topological bond. Next, in case of double-stranded DNA molecule, especially, let's understand this closed. If we talk about closed circular DNA molecule, so most of the plasmids they are there in the closed circular DNA conformation, right, like this. In that case, what you will have is the linking number will always be the integer. In this case, when you have a closed circular DNA molecule, you will have the linking number as an integer in this case, right? Next point is, when you have right-handed helix or when you have left-handed helix, you will have linking number as positive, as negative. So you will see in the later discussion in this particular class, what we'll understand is how we define the right or positive, or how we define the left or negative. But here you need to understand, when the helix is right-handed, then this is the positive, linking number is positive, and when it is left-handed, then the linking number is negative, right? Okay, so now let's move on to another slide and try to understand what are the advanced things in the linking number. Okay, so the next point is the linking number. In this case, what I'm trying to explain, the linking number when you have Nick in the double-stranded DNA molecule, or when the DNA molecule is underwound. If you haven't seen my previous video where I have explained the underwinding, or you can also understand the overwinding stage of the DNA molecule, please watch that video, because we have also explained what is Nick in that case. So here I'll just go ahead and link the linking number with the Nick or the underwound stage. So you can see in this case the DNA molecule is relaxed, and we can take the first example in our case is having the linking number in this case as 200. So we are just taking the random hypothetical condition in this case, that we have a double-stranded DNA molecule where the linking number is 200, right? So if in case of the Nick, and what is Nick? When the double-stranded DNA molecule have that breakage in one strand, that is the Nick, what will happen to the linking number in that case? See, we defined the linking number that once the one strand is penetrating the other one, that will define as the linking number. In this case, that particular definition is, it's compromised, because you have a Nick. Now we can't actually define that linking number. So in that case, what we'll say is, in case of nicked condition, in case of the double-stranded DNA molecule, if it is nicked, we can say in that case, we cannot define the linking number. So we have compromise condition in this case, where linking number can't be defined, but we can define the linking number when you have underwound or overwound stage. So we take the example of underwound state, and what is underwinding? Where that the helix inside the DNA molecule is underwound. So you can say there is a decrease in the linking number, and in this case, I'm taking the example as 198. Let's assume that specifically when the DNA was like that, and due to the underwound stage, two strand they basically made one single twist. So there is a decrease in the linking number, and the decrease, because of that decrease, you have underwound state of the double-stranded DNA molecule, and the linking number in this case is 198, right? So this is what happens in case of the conformations, especially when you have relaxed double-stranded DNA molecule, and when you have nicked, or when you have underwound stage. So now let's move on to the other points in this topic. Okay, so in this case, what we are trying to understand is the specific. Okay, I need to use another marker. This seems like a little bit thicker version of the marker. So specific linking difference, or it is also known as super helical density. And this is, at least you have to remember. You have to remember these things, and it is represented by this sigma here. It's also known as super helical density or specific linking difference. Specific linking difference or super helical density. I just shortened it for you so that you can remember it. Now, what happens in this case and how we calculate this? So super helical density is nothing but the number of turns that are removed. As we have already discussed in the previous case, that when you have underwound stage, that means there is a decrease in the linking number. So here you can see when what is super helical density means how many turns that they are removed from the actual total number of turns that are present initially, So you can see that is why it's known as the difference. So in this case, what we are taking the total number of the turns that were present, and then we are also taking another condition, and which is the difference. So this delta represents the difference, right? So how we can calculate this is LK. You can see the difference. Difference means initially, in case of your reference, right, how many turns were there, and then later on it is subtracted by the result, the final linking number. So in this case, reference linking number and the final linking number that were there after underwinding or overwinding, whatever can happen in that case. That difference is the delta LK, change in the linking number. Right. So if we take the example where initially it was 200 and that it decreased to 198. So in that case, you have 2 as the linking number, and it will be plus. So if we are taking initially we had 200 and then overwinding stage was there, and it becomes, so I think you can't see this. I can write it on the top. So 200, and then the final was increase. So basically what happened, there was increase in the linking number, so you'll have minus 2 as the final delta LK. So your equation becomes sigma is equal to delta LK divided by LK naught, and in this case this is the reference linking number. So LK naught is basically the reference linking number, and LK is the difference in the linking number. We'll take the example in this case, if there is overwinding of the DNA molecule, it will be negative. So minus 2 divided by 200, that is the initial number of turns or the linking number, and then that is the difference. The total sigma becomes minus 0.001, that is 1% change in the total number of turns. That is the definition of this, right? So I hope this is clear, because many times you will find numerical questions coming from this topic where you will be asked to calculate the superhelical density, you will be asked to calculate specific linking number difference or linking difference, and then you have to apply these equations to find out what is the sigma in this case, and based on that you can also explain whether this is going to be positively or negatively supercoiled. All right, let's move on to the next one. Okay, moving on to the next topic, which is understanding the positive supercoiling and negative supercoiling based on the information that we have about the linking numbers. So you can see I've just took the example of the circular DNA molecule, and I have colored them differentially so that you can understand how these molecules, they are interacting and forming the positive or negative supercoil. So as you can see here, you can have positive and you can have negative supercoils. So what will happen in the first case, if you have the linking number that is 200, right? And here, what is happening, there is a reduction in the linking number. In that case, what will happen, first condition is underwinding. You will have positive supercoiling in that case. So that if you have decrease in the linking number, what will happen? You will have that positive supercoiling effect, and as you can clearly see, the one strand is coming on the top of other, and then it is going below the first strand. That is the positive supercoiling. And in second case, if you have increase in the linking number, so what is happening in this case is there is an increase in the linking number, and the difference is +2. In this case, the difference is -2. When there is increase, what will happen? The second part is basically going below the first one, right? And then it is moving on the top of the other one, and that is the negative supercoiling in this case, correct? When you have overwinding effect. So you have two conditions. One is underwinding. Underwinding, we have already explained what is underwinding. Overwinding effect, when you have increase in the linking number, in that case you will have negative supercoiling, and in the other case you will have positive supercoiling. So this is how you can relate it when you have -2, that is underwinding stage, that represents the positive supercoiling. When you have increase, that is the overwinding, and in that case you will have negative supercoiling effect in the DNA, circular DNA molecule. All right, so next is the topoisomers. What are topoisomers? Since we understood linking number in detail, I wanted to include this particular topic so that we can now understand, based on the topological properties, how we can relate two different conformations. And then if you have two different conformations only because of there is a difference in the topological properties, we can call them topoisomers. You can see the example in this case, right? So the definition that I've written here is basically two forms of the DNA, circular DNA molecule. It's basically two forms of the circular DNA molecule that we'll call topoisomers, where there is a difference in their topological property, especially if we talk about the linking numbers, right? And then we can say these are the topoisomers. Simple explanation in terms of linking number. You can see in first case what we have is there is no change in the number of base pairs. Only the difference is in the linking number. In this case, as you can clearly see, linking number is 4. You will have different twists and turns. In this case, you can have different twist and turn in this case, right? So they can also have other differences. Based on that, this property is going to change, obviously, right? So I can explain these two things. When you have difference in the linking number of one circular DNA molecule versus other, in this case you have 4, you have 3. In this case, we can call they are topoisomers of the same the circular DNA, double-stranded circular DNA molecule. So I hope now this is clear. So I hope we have discussed all those points, and then I hope that this is clear. What is linking number? How we can relate the linking number with the positive and negative supercoiling? How we can calculate the sigma, which is the superhelical density? How we can differentiate between the properties of two molecules, whether in terms of linking number, whether it is relaxed, whether it is nicked, and whether it is underwound or super underwound or overwound DNA molecule. So I think we have covered in this particular video quite a lot, and I want you to maybe read more on this particular topic, try to solve more numerical problems related to linking number and also related to the concepts that are there in the linking number topic. For example, the density we talked about, the sigma we talked about, and see how we can how you can solve those, and if you're able to understand those things. If you have any doubts, any questions, you can post in the comment section. I'll try to again explain with the different different information if you're not able to understand. And in the next one, we'll take another topic of this particular chapter, and this is how we're going to follow through to our entire course of the biochemistry topic that we have started, right? So I hope the video was informational and you got the information that you're looking for in this particular topic. And then please stay tuned and do not forget to subscribe to the channel. It helps the channel a lot. So we'll meet in the next video. Till then, take care.